Today, we'll do the second half of female reproductive. We had the first half on the audio lecture last Friday. And then, tomorrow in lab, we'll do female reproductive. And then Wednesday in class, we'll do some eye and ear at noon. And then Wednesday in lab, we'll have our last quiz. So don't forget that last quiz at 2 o'clock. And that'll be over endocrine, male reproductive, and female reproductive. So those are the areas, okay? That's our last quiz. Of course, that's studying for the final exam too, as you're getting ready for those things. So hopefully you'll find that helpful. Then that's it for all the new material. We'll finish up everything on Wednesday. And then the test is, you remember, Tuesday of test week, starting at 8:30. And we'll go through that morning for that. And so that'll be Tuesday of test week. Okay. So, we're really down to the last bit here. So essentially, a week from tomorrow, we'll be done, including the test. But by the end of Wednesday, we will have finished everything new, and we're just studying and working on getting ready for the final exam. So that means our last drawings in the next 2 labs this week, our last quiz this week, and a little bit of questions. There's not a huge number, but a little bit of questions left, and that'll be it. When we do the last quiz and we get that scored and everything, we'll drop the lowest quiz. We'll put the little rule in the grade book, and it'll automatically drop the lowest quiz of the quarter. So that's something to look forward to. Of course, technically you don't have to take the last quiz if you want that to be your drop quiz, but if you say, no, I think I can beat my lowest score, then it's good to take it. Okay, any questions or anything we want to chat about before we get going?
So you said the final is going to be, it's going to have a little bit of emphasis on the most recent chapters, but it's going to be a lot more cumulative than in the past.
That's right. So the reason is we do expand the final, as you know, a little bit more on the digital than usual, a little bit more on the microscope than usual. Written's the same, only the new stuff, and the same length. But although we do the new stuff, there will be a greater than usual number of questions on the previous. And so that is absolutely right. And of course, part of that is we add number of questions. And so that gives us some of that. And then remember on, especially the review, a lot of those questions, not all, but a lot of those questions will be name the slide, or what is this, and so when you're reviewing the comprehensive material, focus primarily on, I recognize that slide, and that's true for both digital and for microscope. Now on the newer stuff that we haven't been tested on, the stuff since exam 3, know that as you normally would. In other words, the specifics within those areas. So we would have, sub areas in there. It won't be just name the slide. There'll be a few of those, just name the slide, but it'll be a little bit more specific. So that's the way I would say to prepare for it. For the comprehensive stuff that we've been tested over before, primarily focus on naming the slide for those. For the new stuff, learn it just like any test. Learn all the ins and outs of it, and that's the same for the written part. Learn it in the more specifics. Is that all right? As far as, and just to remind us, we have 50 on the digital. Usually we have 35, so there's 15 more questions on the digital. And we have 40 on the microscope. Usually there's 30, so we have 10 more. That addition gives us the 25 more questions. So instead of 100, we have 125. The 35, that's the written in there, it's always 35, and that'll stay at 35. So we go, so it might take us, another half an hour longer. As we've been doing them, I think we usually finish before 2 hour approximately, right? Well, this will probably be, I'm estimating a little longer than maybe 2 hours and 15 minutes, 2 hours and 20 minutes. So if we start at 8:30, 9:30, 10:30, we should be done before 11, I would estimate. But a lot of that, some of that depends on you as far as how long it takes you to do the written and all of that. And you shouldn't feel rushed. I'm not saying that at all. I'm just trying to give you an estimate, because some of you may have tests that afternoon or other times, and you may want to know how to plan and whenever you're going to eat and all that kind of stuff. I think if we start at 8:30, we'll be done, I would say no later than 11. I would estimate. That would be my estimate. And maybe a little bit earlier than that. So that's where we're heading there. Okay, well, in the audio lecture, we gave quite an introduction, and we looked at some detail at the ovary, and of course we're going to look at the ovary and the different follicles and the different stages of the cells, the germ cells. We're going to look at that in lab tomorrow. We're going to look at also the corpus luteum and things of that nature. So we're going to see that again in lab. Today we're starting with the duct system, and we'll look at that, and we'll include that duct system, the oviduct, and we'll look at the uterus and the different stages that the endometrium goes through on the monthly cycle, and we'll look at the cervix and the vagina and the breast with active and inactive and that kind of thing. And then we'll look a bit at the structures of early fertilization and some things of that nature. So let's take a look. When we say the oviduct, unfortunately the oviduct, there's two oviducts, they have different names, don't they? And they're all used. We have oviduct, we have fallopian tube, and we have uterine tube. Those are really the big three. They're all the same thing, but three different names. So oviduct, fallopian tube, and uterine tube. The oviducts are going to connect, not quite directly physically, but close, the ovary to the uterus. So when the secondary oocyte is ovulated from the ovaries, it gets swept up by the fimbriae and moves into the oviduct, and then transports it into the uterus for implantation if fertilization has occurred at that time. So this is the ovary up here that we recognize, and here is the oviduct, and you see that it is a couple of different cross sections here as it's kind of twisted there. So this is all oviduct, and this is sort of the fimbriae that sweeps up the secondary oocyte into the oviduct. Here we see again the ovary, we see some primordial follicles, we see a germinal epithelium. This is part of course, the cortex. And over here is the fimbria of the oviduct. We're going to see that the epithelium lining the oviduct is simple columnar, although it doesn't look as typical of simple columnar. It almost looks pseudostratified, but it's simple columnar. And then we have connective tissue lamina here, and then we'll get into some smooth muscle as well. This is from the Loma Linda slides, and this shows you the slide number from the Loma Linda slide set. This shows you the folds in the oviduct, and this is very low magnification, so we can't decipher what the epithelium is here, but it's simple columnar. And then here's our connective tissue in the lamina propria. Now here is a little bit higher magnification, and we're seeing the ampulla of the oviduct. Again, the fimbriated end and the ampulla are outside of the uterus. Eventually the oviducts are going to go into the wall of the uterus, and we'll see that too. But this is the ampulla, which is still outside. And what we see, which is very predominant in the oviduct, is this folding of tissue. And the nice thing about this folding is we can go from the base, like here, and we can trace it, and we can go all the way around and back to the base. And yet I'm tracing it around, you go all the way around, and it comes back to the base. And you can do that wherever you start. Whatever fold, you can go all the way around, and it comes back. And this helps us differentiate the oviduct, for instance, from the seminal vesicles, which have some folds that look kind of similar to this, but you can't trace your way back in that way. So that's kind of helpful. We have the lamina propria under here, and then we get into the smooth muscle layers. There's often 3 layers, an inner longitudinal, a middle circular, like here, and then here's some outer longitudinal. Then we have a serosa made up of connective tissue, and then the simple squamous epithelium of the serosa can be called the mesothelium. And so this is the serosa with its mesothelial lining. These are blood vessels, of course, that we see here, and also in here. This is also a Loma Linda slide, and again, if you can trace the folds. Let's start here, just fun, and we trace it around and around and around and around, and it goes for a ways. We got to go all the way around here, and then we'll come all the way back. And so you can do that. Here's a simpler fold. You just go up and down. So these, this is what it looks like. And here's our muscle layer. And then out here you have the adventitia or serosa, where it's covered by a mesothelium. The lamina propria is this connective tissue right in here. Now we're seeing the epithelium. And when we look at the epithelium, when we take a look at it, we might say, well, is that really simple columnar? Well, it is, but it looks a little bit pseudostratified or stratified, but it really is simple columnar, and we'll show you that on an electron micrograph, the next slide, and you'll be convinced. But just realize it has kind of a pattern that doesn't necessarily look like simple columnar under the light microscope. Some of these columnar epithelial cells are ciliated, the cilia, of course, is very important because the egg, secondary oocyte, is non-motile. The fertilized egg, the zygote, and its structures are also non-motile. So how does it get down the oviduct, down the fallopian tube into the uterus? It's the cilia pushing it along, right? And so there are cases of women who are infertile because the cilia is not properly working and pushing the egg along the way. So, of course, that is important. Then we also have non-ciliated cells, and these are called secretory cells, and they secrete some products, mucus and things, into the lumen. So we have both types. Now, under the electron micrograph, transmission electron micrograph, it's much, much easier to see the simple columnar orientation of these cells. And so they are simple columnar, and we can see that under this aspect. Again, the non-ciliated cells are called secretory cells. They can also be called PEG cells. That's an alternate name. They secrete some products into the lumen. And then we have the ciliated cells. This shows it from the top with a scanning electron micrograph image. And you can see the secretory cells are the ones that don't have any cilia, like these. And then we see very clearly the ciliated cells, which do. So we have that combination. Now, when the oviduct fallopian tube goes into the wall of the uterus, it's called the isthmus. This is another of the Loma Linda slides, slide number 234. Notice that the folds are almost non-existent now, and the lumen is narrowed significantly, and there's still the muscle layer around it as it's going into the wall of the uterus. So we see that looks a little bit different. So this is the oviduct still, but it's the isthmus, or as it's entering the uterus. That looks quite different from this, for instance, where we have all these folds out in the ampulla. Now, what about the uterus? Well, we have different layers of the uterus, and we basically have the endometrium, the myometrium, and the parametrium. The endometrium is the part that builds up on a monthly cycle and then sloughs off. By the way, your wallet's down there, so don't want to lose that. So it builds up. What's that?
Definitely not.
It builds up and then it sloughs off. All right. So we're going to look at that, and we're going to look at those changes histologically. The phases are the proliferative phase, the secretory phase, and the menstrual phase, where it's breaking down. This shows it on that approximately 28-day cycle. Here's the menstrual where it's breaking down, and then under the influence of estrogen coming from the developing follicles, it builds up. We call it the proliferative phase. It's proliferating, and we have some mitotic figures very often in the cells in this phase. And then we have the secretory phase, and the glands, the endometrial glands, uterine glands, often called endometrial glands, they're part of the endometrium. They become very coiled and wider, and that's under the influence of progesterone primarily, but also estrogen coming from the corpus luteum. And so the secretory phase gets the endometrium ready for implantation. Usually fertilization would come somewhere around day 14, plus or minus a day or two, either direction. And then the blastocyst would usually attach about 6 days after implantation to the endometrial wall, and it would usually fully implant at about day 7. So we're looking at, about day 20, 21, something like that. And so we're sort of in the middle of this nice secretory phase. Now, in the absence of HCG, human chorionic gonadotropin, the endometrium will break down. And it'll start to break down probably around day 27, day 28. And then you're starting a new cycle. Essentially, that is the start of the new cycle. So day 28, day 1, and you're starting a new cycle. And that's because the corpus luteum is no longer intact if you don't have HCG, human chorionic gonadotropin, to keep it intact. Okay, this shows a scanning electron micrograph with the myometrium, and here is the endometrium in the deeper layers. Here shows it in another view. And here it is histologically. This is a low magnification. Here's the lumen, and you see the endometrium from about here on, and then here's the myometrium. Outside of that, you have the connective tissue parametrium, which contains, it's really the serosa and the mesothelium coming around there. Now this shows you the layers. So we'll look at this for a bit. This is the smooth muscle layer. Lots of smooth muscle in there. And that is called, the name, myometrium, that muscle layer. Outside of this, we have the parametrium, which again is connective tissue and a serosa. But the endometrium goes from here, where we're sort of tracing on up. That's the endometrium. You can see the line there where it goes. And that's subdivided into different layers. Stratum simply means layer, like strata of rocks or something. Stratum basalis is the basal layer, as you might expect. The stratum basalis is the part that does not slough off, the part of the endometrium. Of course, the myometrium never does either, but the stratum basalis of the endometrium always stays intact. It doesn't slough off. So the rest of it is called the stratum functionale or the stratum functionalis. And you see that here. And it is the part that sloughs off on the monthly cycle. And it is subdivided into the little deeper stratum spongiosum and then the surface stratum compactum. And you have your simple columnar epithelium there on the surface. This is low magnification, so we're not really able to see that. But notice that the epithelium is intact here. As I trace along the laser pointer along here, notice that's intact. What are these structures? These are the glands. We can call them endometrial glands, or you could make it more general. You can call them uterine glands, but generally we're going to call them endometrial glands if they're in the endometrium. They're developing here. If we looked under higher magnification, we'd probably see some mitotic figures. And we are seeing this development of the glands. This phase is the proliferative phase. Sometimes it's called the resting or estrogenic uterus. Why would it be called the estrogenic uterus? Because estrogen from the developing follicles is stimulating this growth and development of the proliferative phase. So sometimes it's called the estrogenic. Now you might say, well, doesn't estrogen, isn't that also secreted from the corpus luteum during the secretory phase? Yes, but also progesterone, and progesterone has a greater impact on it. So this we would just call the estrogenic if we wanted. Because it is only estrogen which is influencing this growth and development, the only hormone. So we have those different phases, and again, we notice that the epithelium is intact. Now when we go to the next phase, this is the secretory phase. We can also call it the progesterone phase, because most of the hormonal influences that cause this phase is progesterone, a little bit of estrogen, but mostly progesterone. Now look at the endometrial glands. They're very highly coiled or twisted spiral shaped, sometimes we call it, and they're quite wide. If we go back and we're comparing, look at they're narrower here in the proliferative and wider here. We get edema, we get this kind of a fluidy kind of appearance in the stroma, this connective tissue. Remember, parenchyma means the functional part of a gland, and the stroma means the connective tissue support. So here, when we're using the word stroma, that's this connective tissue supporting the glands and all of that. And we're going to see that we have lots and lots of spiral-shaped arteries as well as these glands. And so we'll see that. Now, notice here that our epithelium on the surface is still intact. So it's still intact, and that's important because that tells us we're not in the menstrual. We're in the secretory, it's still intact. This is the stratum basale down here. Notice the length of this stratum functionalis. That's going to get smaller when we go to the menstrual because the tissue is sloughing off. The stratum basale will stay the same. Basalis. And then we have the myometrium, which of course will stay the same as well. Here is the menstrual degenerating endometrium. Now, if we look at the surface, look at all this that's breaking up. It's breaking up and sloughing off. Our endometrium is, our epithelium is not intact, and you can see that the stratum functionalis is not as thick as it's breaking off. Here's the stratum basalis, where it gets a little darker in color. So if we go back, we look at this, here's the stratum basalis, and look at the length here, the thickness, and then we go here, the thickness is lessened as the tissue is breaking and sloughing off. So that's our menstrual. So be able to recognize those different phases, and I think you'll be able to do that without too much difficulty. So we want to recognize it as the uterus and the endometrium and the different phases that we see. Now we have the uterine glands, the endometrial glands, as they're also known as, and then we have these spiral arteries, a lot of blood vessels coming in. They start as straight, as you can see here, and then they spiral. And when we look at it, this is a cast of these spiral arteries, and then we took a scanning electron micrograph. Look at all these spiral arteries that we have there in the endometrium. Very, very vascular, as you would expect. It's getting the tissue ready for possible implantation. It has to have lots of nutrition, oxygen, everything there ready and in place. And so it has all these things there. This shows it some more. So these are going to the uterine or the endometrial glands and supplying all that tissue. This is the menstrual phase again. Now when you look at this is a Loma Linda slide number 238. Our biggest clue here, because you might say, well, is that proliferative? Is that, I don't know. Look at the surface, there's no epithelium. And if we see that there's no epithelium, it's got a little bit different orientation. This is the luminal surface over here that we're sort of tracing up and down. But back here, when we looked at it, this was the lumen up here. And if we're looking at here is the lumen. So it's got a little different orientation, that it's, you know, sideways, if you will. But just notice that there's no epithelium, no simple columnar epithelium. That has been sloughed off. And so that's how we can, one of the main ways we can recognize what phase we're in. Okay, well, what about fertilization and development? We won't be doing too much with this, but a little bit. You remember that of the approximately 300 million sperm in a typical ejaculate, only about 100, not 100 million, 100 total, reach the area of the secondary oocyte. And that's why if the sperm count is too low, you don't get enough sperm to the area. If you look, you have the ovum, which is a secondary oocyte. It's surrounded, when it's ovulated, from the Graafian follicle and from the ovary. It has this membrane around it, the zona pellucida, and it has a layer of coronal radiata cells. So the sperm, with their acrosome and their digestive enzymes, have to digest their way through this outer layer of coronal radiata cells and the membrane, and then the first sperm, that the head penetrates the membrane around the egg, the secondary oocyte, the ovum. That causes a clot to all the other sperm, and then there is the egg completes meiosis II, and then the sperm fertilizes it, and it's now a diploid zygote cell. Where does this happen? It usually happens about a third of the way along the oviduct, along the fallopian tube. This shows that information a bit, and here's the egg undergoing meiosis II, and then fusing to form the zygote. So it usually happens, maybe about a third of the way along. Here's the fimbriated end of the oviduct to the fallopian tube. Here's the ampulla, and then it goes into the isthmus right there, into the wall of the uterus. And so once the zygote is formed, about 30 to 36 hours after, it starts dividing by mitosis, and you go to the 2-cell, 4-cell, 8-cell, then a ball of cells. The more you love, then it turns into the blastocyst, and that's what gets implanted again, it attaches to the endometrium about day 6 after formation, after fertilization, and it gets fully implanted about day 7, a week after. So these are the stages that we see. Now, when we look at the blastocyst, we have a couple of structures. We have this area in the diagram, that's called the inner cell mass. We see it here, there, and here it is. The inner cell mass, these cells are what is going to form the baby, the embryo, the fetus, baby. The outer cells on the very outer edge are called the trophoblastic cells, and they form the auxiliary structures, like part of the umbilical cord, part of the placenta, and that kind of thing. So the supporting structures. Now, some of this is done with maternal tissue, and so there's a combination of the two. This is the endometrium here in our diagram. It's attached, and it's going to get fully implanted into the tissue of the endoderm, of the endometrium. The trophoblastic cells, by the way, early on are also the cells that produce HCG, that signal to the mother's body that she's pregnant, that causes the corpus luteum not to break down after about 12 days of formation, and it causes the endometrium, therefore, to stay intact because progesterone and estrogen are still being secreted from that intact corpus luteum. Histologically, this is what this blastocyst looks like. These are your trophoblastic cells out here, coming around, and this is your inner cell mass. Of course, it's not really floating like this. We just, in the cut, we don't see the cells all the way across. But this would be our inner cell mass cells. So this looks so nice diagrammatically, it actually looks more like this.
And that's implanted?
This is implanted. So this is the surrounding endometrial tissue. Okay, eventually we form an umbilical cord, and we'll be looking at that under the slides tomorrow, so we show it here. We have a placental vein and some placental arteries. This is the mucous connective tissue, or embryonic connective tissue, we can call it. The allantos eventually becomes the yolk sac, forms the blood cells, and that kind of thing. That also is where the immature germ cells for females come from the yolk sac. But anyway, you have that. Now, the placenta is this helpful endocrine structure that's in the uterus with the baby, with the fetus, and it comes partly from the baby's tissues, if you will, and partly from the mother's. And so they fuse to form the placenta. So we have maternal tissue over here, and we have that. Now, what is that? That is part of the endometrium of the mother. And then over here, we have this trophoblastic cells that are developing from the baby, if you will, and they're going to all merge to form the placenta. Now, when we look, this is a lower magnification here, this is a higher magnification here. We have this chorionic plate of the placenta, and then we have these chorionic villi. These chorionic villi are simply these finger-like projections of the chorionic plate of the placenta. And so here they are, these finger-like projections. Now, when we blow that up, we can see that these epithelium is of two cell types. The one right next to the lumen is called the syncytial trophoblast layer, and the deeper layer that's a little more pale staining that we see here is the cytotrophoblastic layer. That's this layer. So we'll see that, and that makes up, again, the placenta, the choroid area that we see there. So we'll see that on the microscope tomorrow as well, and we'll identify the different types of these two different cell layers. This is from the Loma Linda slide, and this is this chorionic villus, finger-like projection of the placenta. And again, you see the two layers here. This is fairly low magnification, but you can see the two cell layers along there. And this is mucous connective tissue, embryonic connective tissue, if we want to be a little bit more general. And that's what that is.
What cell type are the two layers? Are those columnar, or?
They tend to be more cuboidal,
Okay.
but it somewhat depends on how far along you are.
Okay.
Because if you look here, they're probably a little more flattened. And then they get a little, whoops, the wrong way. They get a little a little more rounded up. Now, of course, the uterus narrows down to a narrow neck called the cervix, which then is continuous with the vagina. And this shows the cervical lining. Again, it's simple columnar. They secrete a lot of mucus. There's mucus glands in the area as well. Here's your connective tissue, lamina propria. And this is the cervix. Now, here's some mucus glands. Look at how pale staining the cytoplasm is there. There's the nucleus for these glands. And you have your connective tissue. There's usually, quite a few lymphocytes, lymphatic tissue there as well. Here's a simple columnar epithelium. And then you come to the endocervical junction. So this is again is a Loma Linda slide, and you see it going from simple columnar as we're tracing it along, and fairly abruptly into stratified squamous of the vagina. The epithelium here of the vagina is stratified squamous, parakeratized, so the nuclei come all the way to the surface, and so that's something that we'll take note of. Again, some lymphatic tissue and some endometrial glands or intracervical glands right here at this junction. Those are mostly mucus producing, and so on both sides, here we see the lumen, and here we see the transition at this junction. This is an important area medically, clinically, because most cervical cancers occur right near this junction, the majority of them. So this is a place they often look. This shows it in a lower magnification with the sort of the abrupt transition here, and here it is in higher magnification. And again, stratified squamous, parakeratized, and here's simple columnar on this side. This is now the vagina, and again, stratified squamous, but notice the nuclei come all the way to the surface, so we classify it as parakeratized, and the nuclei are retained all the way to the surface. This is a different kind of stain. It's not H&E. You can see just a little the structures here. Notice it's quite a bit of blood vessels, veins, sinuses, as well as the collagen and supporting tissue there. Here it is again. You have the lamina propria and submucosa. Down here you have the muscular layer, and that's smooth muscle, as you would expect. Ooh, we're almost there, aren't we? All right, so the breast, we have the resting adult breast. This means it's not producing milk, okay? That's what it means there. And so these are lobules, and these are the glands, which are going to, when they become activated, produce milk. And we have the connective tissue in between, and then all of this connective tissue, and then we have lactiferous ducts, and fat is the adipose here. So this is an adult breast. A senile breast, meaning from an older person, would have a higher proportion of adipose. That's really the main difference. Now, what about an active breast? This shows, look at all the glandular material here, these alveolar glands, the lobules. This would be loose connective tissue in between the lobules, and this would be more dense and regular out here. And these we would have lactiferous ducts. That's going to carry the milk to the nipple, to the baby, and so we see that shown here as well. So this is, you first produce colostrum, which is this protein-rich, thick, secretory product, and then it turns into, eventually into breast milk. This shows it on the outside. And this is a Loma Linda slide showing the nipple. It's a special type of skin, and it shows the keratin here. So we see it's the skin, and this is a nipple showing the lentiferous duct coming up, and the smooth muscle. This is the dermis area and the epidermis. So we have the epidermis and the dermis. So it is skin, but it's a modified skin. Oh.